Oh, okay. I can take the jump drive, though.
Well, we'll put it right on there.
Okay, that's fine.
I'll just put her talk's already on there.
Oh, okay, great. Here, you have a minute or two because we're just getting settled, so.
Okay, so there's that one gone. Okay, that's that one. Tidy up the desktop while I'm here.
Also, just be aware, I think these are all running at the same time. So we've got a 4:30 DADS panel as well. So if that's something that's of interest to you, please feel free to join that as well. They're going to have alcoholic beverages, so prepare yourself. So DADS panel, CDKL5, I think they're doing that in the Cafe Lounge with Rick Up. Good? Oh, okay. Very good.
Okay, this is there. Okay, so that's cool. I'll just get rid of this. And I don't know what this is. Do you know what that is? Okay. There. Okay, so it's done. Good. So apart from that, cool.
Were you able to get it on? Oh, good.
Yeah.
Okay, great. Okay, great. So this is our 4:30 session, Developing Biomarkers and Outcome Measures for CDKL5. It's actually a two-part session. We're going to talk about using brainwaves as a measure of a drug's impact and the implications for CDKL5 Deficiency Disorder. And then the part two of this is the quality of life domains, important for individuals with the CDKL5 Deficiency Disorder. And looking at quality of life as an endpoint measure for clinical trials. And our speakers, Jenny Downs from Telekids Australia, will be doing part two, right? And Eric, who you've seen before from our epilepsy panel with Tim Benke, is going to be doing part one.
All right. Thank you all for coming to this session. It looks like we've lost lots of parents. And this was more towards parents. But all right, we'll go anyway. So I was going to talk to you all about developing a biomarker, and particularly an electrophysiologic biomarker. And that means a biomarker that records brain waves as a way to determine how a disease progresses or a response to a treatment. And for those who don't know what a biomarker is, usually the first thing you do is you search Google. And if you search Google, this is what appears as the first hit. If you first Google is the definition. Google nicely now gives definitions. And so a biomarker is a measurable substance in an organism whose presence is indicative of some phenomena such as a disease, infection, or environmental exposure. Biomarker is a portmanteau between the words biological and marker. So that's a good crossword word for you if those who don't know it. And they're getting a lot of attention now to develop biomarkers for diseases. And this is something that's very important in this space. We'll go to that second. Just my disclosures quickly. The biomarker study that I'm going to talk to you about is funded by the NIH, Penn Orphan Disease Center, Lulu Foundation, and RettSyndrome.org. And as I already said, I serve as a PI on a number of drug studies. So what I was going to try to do today was give you a brief overview of how clinical trials lead to new drug approval, because this is why we want ultimately why we want a biomarker, because we want new drug approval. And looking at in the audience now, I think most of you are going to know this, so I'll go quickly. And then a brief overview of what biomarkers are. Is a good number of? Okay. Good. Okay. Perfect. Okay, perfect. And then discuss kind of what we're studying, what are brainwaves, discuss the data that we've been collecting and what it's starting to show us, and then discuss how physiologic biomarkers can help CD trials advance. So clinical trials, this is from a think tank that did a study of over 1,500 parents, and the vast majority of them do not understand how a clinical trial works. So this is something that's important because people are being recruited into clinical trials, they're consenting for clinical trials, and I think a lot of people who are consenting don't really truly understand what they're consenting for. The discussion of what is informed consent could be a whole other day, so we'll skip over that now. But most clinical drugs progress through a standard set of states. The first state is the preclinical research state, where the drug is first tested on a number of conditions, or a condition in particular. It then moves through the clinical, if it passes that preclinical trial state and there's efficacy in a model system or some other way to test that this could work on the disease of interest, it goes to clinical trials, where there are 4 phases. The first phase is typically a safety phase. The second phase is typically safety and dose finding phase. Phase 3 is the gold standard. It's the placebo-controlled clinical trial phase, which is looking for efficacy to show that the drug works compared to a placebo, which is gold standard now. And many people are questioning, in rare disease space, do we need phase 3 clinical trials? How else could we potentially approach this? Because when you have so few patients, using a standard pathway might not be the way to go. And Dr. Kessler talked about this earlier today. And then there's phase 4, which is extension phases and looking for more safety and efficacy data. And then there's post-marketing surveillance. So once the drug is out there, the FDA continues to look at the drug to make sure that other things don't arise. And the best example of that in the space you might all be aware of is felbamate. Felbamate made it through clinical trials, or felbutol made it through clinical trials, and then afterwards it was found that there's a high incidence of liver failure and aplastic anemia, which made it get reassessed on its safety profile. And this process is rather expensive. So if you forget the preclinical research phase, which takes a lot of money in itself, when you get to the clinical trial phases, these in standard drugs for more typical non-orphan disease space, these are very expensive. So, a phase 1 trial could have up, 20 to 100 healthy volunteers, and it could take a couple years to go through the phase 1 trial. A phase 2 trial usually takes several hundred volunteers, and it could take a few years. You get to phase 3, depending on what your endpoint is and how much of an effect. It could take, you can have up to 3,000 or more patients in a trial, and this can take many years for a trial to progress. With the fastest trials, so the Greenwich Pharma's first CBD trial in Dravet, they enrolled that trial in 6 months, which is like unheard of how fast they enrolled a trial. And then it gets to the FDA, where you submit a new drug application, an NDA, and that, depending on the status of it, could take up to a year for them to review. And then to get out into the market, it could be even more time. So this whole process can take years and years and years to go from preclinical work to approval of a drug. And so the question is there ways to expedite this? Are there things that we can have and use that can get through this with less money, less time, and improve the outcomes of health in patients, and in this case, in CDD. And this is a slide which is from a very nice review from Liz Berry-Kravis, who's in Chicago, which was done with there's like 15 authors on this paper, both from academia and industry. And this is actually a list of all the drugs that have been tried in Fragile X, and all have failed. So Fragile X is a more common neurodevelopmental disorder, and they've been working on trials in Fragile X since 2002, and nothing has hit the market. And they've all failed for a variety of different reasons, and this review nicely goes over those reasons. And one of the things this review really discusses a lot in it is to talk about the importance of having outcome measures that really match your disease and having biomarkers that could be used to appropriately track the condition. And that we, in this neurodevelopmental disability space, don't really have that. And Fragile X jumped into these trials without having the appropriate outcome measures. And so some of the reasons that their studies may have failed is that we weren't measuring the correct thing. And so if you don't measure the correct thing, you're not going to get the result that you think you might get. Of course, some of these drugs could have failed because they just weren't any good, but the paper doesn't really talk about that. So that brings us to, you have all these failures. Is there something we can do better? And this is where biomarker concept comes in. And I already gave you the Wikipedia definition, or sorry, the Google definition of biomarkers. This next definition is from a review in the current opinion of HIV and AIDS research. So this is not something that's just in the neurodevelopmental disability space. This is across medicine, coming up with better biomarkers. And what's important is and then the next definition is from the FDA, directly from the FDA's website. And then biomarkers don't have to be clinical endpoints, right? They can be a surrogate endpoint. But this then and a surrogate endpoint is a substitute for a clinically meaningful change. So it's something that you could see without actually getting to that clinically meaningful endpoint. And, but this is something where if you're on a regulatory issue becomes important because the FDA has to agree to your biomarker as an endpoint in the study. But so biomarkers can be used for many things. And so there are many different types of biomarkers. So there are biomarkers that are natural history markers. So these are markers that precede or report out the progression of disease. And the best example of this is probably in diabetes, which is measuring A1C, hemoglobin A1C. And the level of hemoglobin A1C does a very good job of telling you how well treated or how progressed a person's diabetes is. Then there are drug activity markers. These demonstrate that a drug is engaging its target. And this is a very important thing in the neurodevelopmental disabilities. You're giving a child a drug and say it's not for seizures where you're counting seizures, but you're giving a child a drug and you're looking for a change in behavior. And how do you know it's actually working on their brain? How do you know it's getting to where it's supposed to go? And so drug activity markers are markers that demonstrate that a drug is engaging its target. And so an example of this is that for everyone whose child has been on a benzodiazepine like Valium or Transine, clobazam, beta activity, fast activity appears on the EEG. So you could use the presence of beta activity as a marker that your benzodiazepine is working. The fact that your kid's falling asleep is another marker that it's working, but this would be a biomarker that it's working. And then surrogate markers, I already mentioned. This could tell if a clinical purpose endpoint is being reached. And an example of this is for cancer, is tumor recurrence and MRI findings. So that the fact that the tumor has gone in MRI, you don't actually know if the tumor is completely gone. You're not in the person's body. You don't see if there's still tumor cells there. But it's a surrogate endpoint for resolution of cancer in a patient. So it's a good surrogate marker, and that's what's used clinically in most cancer studies. And then finally, predictive markers. These are markers that will predict if a treatment will work or tell about the severity of disease. So do you have something at time A that will tell you what time B will look like? Or do you have something at time A will tell you that a drug will work or not work? And those are predictive markers. And that is, ultimately the goal of, as a physician, I would love to have someone comes into my office, you send a blood test, you do some sort of test, and you say, here, you need this drug, this thing, and this is what's going to happen to you, and takes all the guesswork out of it. Make my life a lot easier. I could spend more time drinking beer. No, actually not. That's not true. But that would be a good thing to have, and it would be an important thing to have, because as you're all aware, treatment of seizures is a trial and error process, and we're not very good at it, and we just do our best. So it would be good to know that patient A is going to respond to clobazam, patient B is going to respond to this drug, and that would be a very useful thing to have. So how does one develop these? So biomarkers, are particularly unique. There are some unique features of the neurodevelopmental disabilities that really lend itself to using biomarkers. Because in these conditions, there are both acute symptoms to treat, like seizures or lack of sleep, and then there's the chronic condition that is the encephalopathy, the continued change in the development and their function that needs to be altered. So you have both drugs that you want to treat something acutely, a specific symptom, and you have drugs that you might want to disease modify. And this is something that Dr. Demarest was mentioning early. And then for the latter, the time to reverse or improve a disease is not really known. So for asthma, you give someone a drug, the person has spring, there's allergies, they have 3 asthma attacks a month, you can kind of say, we're going to treat for X amount of time and know what happens. But for a child with CDD, how long do you need to give a drug before their sleep improves? How long do you need to give a drug before they become more alert? What is the time that we need to treat? We don't know. And people are are designing clinical trials based upon kind of standard approaches of using 12 weeks or 24 weeks to do this, but this is just random. I mean, there's really no data to support how long you should be giving a drug before you expect to see a response. And some of these things might fail because we're doing it for way too short than that needs to be treated for. And then you have multiple brain regions involved. So if you have a drug that you think might work on improving gait, that might be targeting the basal ganglia or the cerebellum or the spinal cord, whereas cognition, it's maybe frontal lobes or parietal lobes. So it depends on what you're looking for, and so you might be able to develop a marker that can piece out from the global picture how there's change in a particular area. And so that's something that could be relatively unique to the neurodevelopmental disabilities. These are small populations. As a whole, the NDDs are, up to 1% of the population. But any given etiology, any given particular NDD is a very small incidence. They're all orphan diseases. They're all less than 50,000 people. And these typically are fragile populations. These kids aren't the healthiest kids in the world. As we just, they have GI problems, respiratory problems, aspiration. So to expose them to trials, to drugs, is a problem, and it's something that needs to be think about. So if you can develop a marker that would limit the amount of exposure or predict how something's going to be done, you can really improve the way a drug gets through or a treatment gets through. So with all this in mind, one of the things to look at in a neurodevelopmental disability are brain waves, right? Because these are all disorders where the dysfunction is primarily in the brain. So if you want to study brain waves, you have to measure it. So this is just an image I pulled off the internet, but it's a very nice image because it shows you both the spatial and kind of temporal resolution that you can use to measure brain waves. And so you can stick an electrode, you can stick an electrode, it doesn't show up again. Twice it doesn't show up. You can stick an electrode in the brain, which has a really sharp tip, a 3-micron tip, and record from a single neuron. Now that's not something you're going to do in a person, because to just expose their brain to record one neuron is probably not worth the risk-benefit. But it's something you can do in a mouse or in a primate that you can record. And next would be recording from only a few hundred neurons or a few thousand neurons. And that's using these what are called MEA or microelectrode arrays. That looks like a little bed of nails torture chamber, but it's actually being used in clinical practice. It's called the Utah array. And in patients undergoing epilepsy surgery, this can be implanted into the superficial layers of the cortex and record from hundreds of neurons simultaneously to actually look at the activity of multiple individual neurons. Because right now, most people are looking at the whole brain's activity, but what's happening on the cellular level, and that's something that this MEA can do. But again, it's limited because it requires brain surgery, it requires a device that actually goes into the tissue and potentially injures the tissue, so you only want to do it in tissue that you think is going to come out after like epilepsy surgery. The next is ECOG, and this is EEG, but sitting on the surface of the brain. So the electrodes are right on the surface of the brain. And the problem is your brain generates electrical activity, and I'll go into this in a second, but that electrical activity is really, really tiny. And these amplifiers and the systems we use amplify that activity so we can record it. But when you do it on top of the skin, there is layers of tissue it has to go through that makes the activity even harder to record. So if you can put the electrodes directly on the brain, what? 4 more minutes? Okay, ooh, well, gotta go faster. Then you can record better. So these are all the different ways you can record. And this is how brainwaves are formed. So I'm gonna skip this because of time. And so in CDKL5, there are a few papers that actually have reported out actual EEG data. A lot of papers talk, write what the EEG shows. A few actually show it. And you can see that they're at different ages. So this is from 2 different papers, which have different patients at different times, that the EEG can change over time. The EEG can have very abnormal activity, have slow spike wave, a lot of different changes. And so you need to know kind of where the state is of the patient in order to look at different activities. Or are there measurements that you can make regardless of the state that will tell you something about the underlying brain function? So in CDD, why is the brain dysfunctional? Well, because in this condition, it's probably synaptic cellular dysfunction that's causing individual cells and then groups of cells to not function properly. And so we can potentially use EEG or evoked potentials to measure that synaptic or cellular dysfunction in a way that you couldn't clinically. So how does one do this? So an auditory evoked potential, or an AEP, a visual evoked potential is a stimulus where you provide a checkerboard or different stimuli, and you give a couple hundred stimuli to the patient, and you record brain activity. The AEP, you provide a auditory stimulus, which I don't know if this is playing. No, it's not loud enough. But there's a white noise stimuli that you can present, and then you can record the brain system using standard EEG systems. And so is this methodology robust? So if you do this at Denver, or if you do this in Boston, or you do this in Philadelphia, do you get the same activity using different systems, using different times and places? And so we tested this by sending Dr. Tim Roberts here, who's the Director of Radiology Research at the Children's Hospital of Philadelphia, as a human phantom. So he went to 4 different sites and got a AEP and VEP at those 4 different sites using, as you could see, different electrode systems. 2 of them have the same amplifiers, but the other's different. And if you look in the middle, his evoked potentials are the same no matter where he is, when he is. So it's a robust system. And if you actually quantify that, the latency of the response and the amplitude of the response, how tall it is and how fast it is within a millisecond of each other no matter where he is. So it is a robust system. So these are robust measures which could then be good to use in clinical scenarios. So the other nice thing about some of these electrophysiological biomarkers is you can go from model to human. And so these are papers published, both actually out of Penn, out of Joe Zhao's lab, looking at, this is in the MECP2 mouse, looking at the mouse data up top, a mouse EEG up top, and then on the bottom is a mouse auditory evoked potential. And this is a different MECP2 mouse out of Boston, also looking at auditory evoked potentials. And this is a nice paper which was published in 2015 where they compared the mouse auditory evoked potential to the human auditory evoked potential and found very similar changes. So it says that not only is it robust from place to place, but it's robust across models. So that would be a really nice thing to have to test the drug in an animal and then look at its response on an AEP and then do the same thing in a person. And this is this similar study also out of Joe Zhao's lab in the CDKL5 mouse looking at the AEP as a biomarker. And then there's been a few studies doing this, all with relatively small numbers of patients. So this is from that same Boston paper looking at auditory evoked potentials. The top is from typically developing children. The bottom is from girls with Rett syndrome. And you see the waveforms look very different. And if you quantify that, there is significant differences between typically developing controls and Rett girls, both in the auditory evoked and the visual evoked potentials. What's interesting is the visual evoked potentials were more abnormal, but as many of you know, Rett girls actually are very visually attuned. So why were their visual evoked potentials done? So other studies have been done. And so what we've been doing recently, just to show you a little bit of this data, is to try to get a bigger sample from across multiple sites to look at these evoked potentials. So the Rett Natural History Study has been doing this biomarker study. It's called 5212. If any of the sites that you're involved in ask you to participate, that would be terrific. And what this requires, so we're looking at these same markers that they've looked at previously. We have different outcome measures. It requires that you have MECP2, CDKL5, and these inclusion criteria. And we're recruiting up to 30 CDKL5 patients, which include 12 additional ones that are funded through the Penn Orphan Disease Center, and controls to match to those patients. And so we're starting to have results. So we've actually up to, across the board, I think we're up to like 60 patients now. There's more than 6 CDKL5 at this point. And if you look compared Rett versus MECP2 dupe versus CDKL5, FOXG1, and controls, CDKL5 has a different morphology than the Rett and the FOXG1 and the others. What's interesting is it's bigger. So in the visual evoked potentials, they have a bigger response, which is very fascinating because as Dr. Demarest was talking about earlier, these children also are thought to have cortical visual impairment. So why do they have such robust responses? Now, a robust response doesn't mean a good response, but it's a different response. And this is the quantitative analysis of that. And if anyone wants to see the data, I could show it to them afterwards. And this is the auditory evoked response. We're having a little bit more trouble getting really good auditory evokes than visual evoked responses, but we're getting ones that we see are reasonable. And this is at the data. And again, they do look different than the controls. So there's clearly a difference in the cortical responses to both these stimuli in these patients. This is the quantification. So each disorder seems to have its unique evoked potential profile. There's some consistent results between humans and mouse models, and we can reproducibly do this. So this might be a potentially very good method to use as a biomarker, though there clearly are challenges that we're still overcoming in this process. And there are many other biomarkers that people can use, and these are just examples of some. And just for one biomarker, for the families who are here, that Boston Children's is actually collecting saliva for, there they are, they have a table out front. They're collecting saliva from patients and questionnaires. So if anyone's families are interested, they have IRB consent. You can enroll and be part of a microRNA biomarker study today or tomorrow. So please participate. And we'll just end with that. And so thank you all.
Shall I get going? Great. So I'm talking about CDKL5 in relation to the very important outcome of quality of life. I'm sorry, the wrong one. Right, so in the first instance, I know that I'm one of just 2 or 3 people who are from Australia, so I can show you the city, Perth, where I'm from, and also introduce you to our very unique wildlife. Why we have black swans, I don't know, and also our beaches and the very hauntingly beautiful interior that is as dry as you could imagine. But also show you the team that Helen and I work with, and you can see a dozen of us or so who worked with us very thoughtfully and very passionate on all of our projects. So this is one of Helen's slides, and I show this because it's showing us that it's only since 2004 when we recognised CDKL5 as a distinct genetically caused disorder. There's a genetic cause for some of the children's symptoms. And here we can recognise that over just 14 years we have made extraordinary progress, but that's because of people who have gone before us. And I also show this picture of these 2 girls, 2 twin girls, with, of course, within one family, but they demonstrate how variable the phenotype is. One who has symptoms that overlap with Rett syndrome, and the other girl who was diagnosed with autism. She is extraordinary. They are 2 twin sisters who are extraordinarily different and have that same mutation. And so phenotype variability has been looking at us for a very long time. So these are the main symptoms of CDKL5, and as you know, it's a serious disorder in terms of seizures and global developmental delay. But I would also like to highlight that there is a very parallel gestalt, and that is the capacity of the children to learn new skills, to enjoy participating in activities, to have a winning smile, and to establish and maintain relationships. And it's by recognising these other qualities of the children that we can really start to put together the need for us to better understand the idea of quality of life, either that is effective because of the health and disability issues, or can still be very good despite the health and disability issues. So quality of life is your feeling of satisfaction with your overall life experiences. We all know intuitively what quality of life is. It is an extremely neglected area in CDKL5, and it really would be an important outcome measure for us to be better on top of when we're wanting toolkits with which to select the best measures to measure the precious treatments that are going to come our way. And of course in CDKL5 the effects are very pervasive in the children's health, their behaviour, activity and their participation is really restricted, and so we really need to take the child's whole life into account. So I would like to introduce you to our initial quality of life work. We started with no well-validated measure for children with intellectual disability with the laying of a grassroots foundation. We recruited children from across Australia from 4 different diagnostic groups: Down syndrome, autism spectrum disorder, Rett syndrome and cerebral palsy. And there is overlap in the symptoms, not the cause, but in the symptoms with children with CDKL5, except for Down syndrome, but for the other 3 groups. And from all of those disorders, we conducted a total of 77 interviews, and there we collected really strong, rich stories from the families, and we were able to code parental observations of their children. And what we found were domains of quality of life, not surprisingly, that matched to health and wellbeing, to activities and to immersion in the community. And what we also found was that those domains were consistent across each of these 4 disorders. So I would like to just show you one example and a quote from the mother of a young or a teenager with Rett syndrome, and that was the natural environment and how important that was for quality of life. And this mother, in the quote, which you can read while I speak, is talking about her love of the outdoors, her love of that sensory experience from nature, and a picture of another young girl surfing and having a very, really enjoying that. And I felt very, we presented this at an international conference in relation to quality of life, and people who are the fathers of quality of life in relation to intellectual disability said to us, how did we miss that? And I pay tribute to the stories and the time parents spent with us to sometimes speak for an hour or an hour and a half in each of those interviews to give us very observable information from which we could create our framework. So we extracted from all of those interviews, 77 interviews, 77 statements, and we worked as a team to workshop those items backwards and forwards. We then came up with 51 items, which we felt were applicable to all of those children. And we then submitted those 51 items to a process called Think Aloud. And this is the equivalent of another technique called cognitive interviewing, and it's what the police use when they're working to really understand evidence. And that's what we used with our draft measure. And by listening to parents talk our items out aloud and explain why they would choose them for their child, we were able to use their words in the items. So our scale now speaks their language. And so that was a really a positive thing that our scale was able to pick up. And we could also find out items that were duplicating ideas. We didn't necessarily see that, and that didn't make sense. So we were able to prune it significantly down to 43 items. So we then put the 43 items through an exploratory factor analysis. We then submitted it to a confirmatory factor analysis to check that the scales loaded. We piloted it with 253 families across those 4 disorders, and then with our factor analyses, we came up with a 6-factor solution with just 32 items. So one domain is social interaction, one is leisure and the outdoors, one relates to physical health, one relates to negative emotions, very challenging behaviours, one relates to independence, which might be helping with tasks. That doesn't mean total independence. And also another subscale is about positive emotions. So we've done a lot of other statistics that I'm not presenting today because of time that looked at the convergent and the divergent validity, and the Cronbach alpha values were suitably high. So that sort of support is important for the validation. But we also compared the scores across the 4 groups, and using Rett syndrome as our baseline, we found that the children with Down syndrome had stronger social interaction scores, independence scores, and better physical health scores. And not surprisingly, whilst the children with autism had better independence scores, they had more difficulties with social interaction. So these sorts of ideas are consistent with theory and help us to understand how this scale is performing. So we now want to see how we might be able to relate it to CDKL5. And so what we have done is a qualitative design. We're getting a lot of experiences with qualitative studies, and here we were able to recruit children from the international CDKL5 database. We sampled purposively so that, as you can see on the right side, we had a range of characteristics by age, by walking, by communication, by whether or not the child has a gastrostomy or not, and boys and girls. So we selected so that we could capture a range of experiences. We did semi-structured interviews, we transcribed those, we did all the methodology that goes with the qualitative study. And then we used all of the data and we mapped it to the framework that we identified for Rett syndrome. So we saw, does it hold for that same framework that we had previously found? And this is just to show you, now we're thinking CDKL5, what our interview questions were. This was our schedule, and you can see there are 5 questions there. And that was enough for families to talk to us for sometimes 1.5 hours about the things that were important for their child and the things that were difficult for their child, and illustrate with that. We asked, how do you know that? What would I see if I was there? And so those were the observable behaviours, and those were the behaviours that we coded, not a parent saying she loves being with her family. We would say, well, how do I know that? And so we would code what we would see. And we feel that that's a really important part of the methodology, because the children are not able to speak for themselves on this very important personal issue. And this is our way to maximise the content validity for the children. So it's one day with better communication techniques, we might be able to ask these more complex questions, but at this point that was an important strategy that we implemented. So we found these were the domains that we mapped across to, and we found exactly the same domains of quality of life for CDKL5 as we had found for the other diagnostic groups. And I will illustrate some quotes from 3 of those domains. For example, physical health. On the right of each of these next couple of slides, you will see the elements within the domains from the Rett syndrome data. And on your left, you can see a quote from our CDKL5 data, and you can see how richly they demonstrate that particular domain and that aspect of that domain. So here we've got a child who was having so many seizures on her holiday that she was sleeping through the whole time. And this was her holiday was spent sleeping, and that was an important challenge for that child's potential to enjoy that family time together. For communication, we have the importance of communication aids, and this is a really very lovely quote from a grandmother who was, a mother who was also with her grandson at the time, and the daughter using her iToby device to actually tell the grandmother, her mother and her nephew, I think I've got the family connections right, her nephew to stop talking, I want to get on with my meal. And she was satisfied that the daughter at the end said, "That's so cool." She was satisfied that she had communicated her message very clearly within the context of a 3-way social setting. And here, social connectedness, a young man with CDKL5, and he's going into a shop and seeing a shop assistant that he hadn't seen for some time, and there is recognition, there are greetings, there is happiness and joy at this interaction, and this is exactly what we have seen across the other disorders. So we can say from a quality of life point of view that each of the same aspects are important across different disorders. And this is important because it helps us to understand whether the quality of life scale might be appropriate for CDKL5. We also noticed when we divided the data set up into the different age groups that each of the domains were represented in preschool children, school-age children and adults, and that helps us to feel that we can use this scale across a wide age range in CDKL5. So now if I could just show you a couple of little sections of the questionnaire. The health and wellbeing domain has 4 items, and you can see they are parents' words as we worked through Think Aloud, and these are the 4 items that were maintained, and we're rating them on a 5-point scale. And for example, here is a selection of items from the family and friends. It's social interaction. We've structured the name of the items, the domain slightly differently to make it more family friendly rather than a medical sort of domain name. So overall, I think what has been an important lesson for us as we've been working through these processes is the frameworks that we have worked with. We've worked with a conceptual framework and a validation framework. Conceptually, we have worked faithfully to models of quality of life and not been deterred from that. Quality of life is equally important to children with a severe disability because they have so little voice in the literature, and this is a really important task to provide them with more voice in terms of what's meaningful in life. So we have stayed really faithful to fundamental definitions and also the ICF model in relation to disability. And we were very cognizant of how we worded the questionnaire. We worked with families to find how we write each of the items, and we've framed it in a positive framework, well-being, not ill-being, except for the negative behaviours items, because parents really wanted to acknowledge those up front. And it looks like you're kind of shoving them under the carpet when you're asking about never has a challenging behaviour rather than they do have a challenging behaviour. So it felt more honest to refer to that in the negative direction rather than the positive direction. And we felt that was also important because when you're talking about quality of life, everyone needs to be protected and have their self-esteem taken into account. So we've really felt that this needed to be a very consumer-friendly scale. And secondly, the psychometric modelling. So we've had appropriateness to purpose and its feasibility very robustly tested by its grassroots foundation and working with the validation across a very large number of families so far. And we have done some of this looking at its reliability and its validity. So there's still a lot of work to go, because that is the business of developing a scale and then knowing that it's working across multiple settings. So what is next? We hope that this will provide greater capacity in clinic to more clearly identify support needs. We know of a couple of families who have used this model to help them in identifying what funding needs they are needing in Australia. So that's a model that parents can work with and not feel deterred in asking for all these things rather than just one or two. And it has empowered this mother who has used this model. And it could also be an appropriate outcome measure for clinical managements. In our immediate research world, we are implementing it within a larger questionnaire looking at the determinants of quality of life, and we also have included it in our follow-up CDKL5 questionnaire. And we hope to understand how this measure is performing in CDKL5 in the very near future. And I'm really excited that it's an exploratory measure in the upcoming ganaxolone trial as well. So I think that's all. Thank you especially to all of our families who participated, the 77 who gave their time with and their heartfelt experiences in all of the interviews was wonderful, and of course all of the families who have been piloting the study. The quality of life interviews with the CDKL5 families were conducted by an honours student that Helen and I were supervising, and she really picked up on understanding how to connect with these mothers and fathers, and really took with a passion and exhibited a lot of thoughtfulness when she was talking to those families, and the data was extremely high quality. And of course the foundation who provide our database with some ongoing support. Thank you very much. Sorry, I'm early. You're done.